Good evening, everybody. Now we are going to discuss about biological tools for the recombinant DNA technology. That is for cutting the DNA, removing of the DNA, and recombination of DNA. There are the different biological tools which are used to produce or to construct the recombinant DNA or the chimeric plastid. First one is the enzyme, cloning vector, and component or the component host. So these are the different biological tools. With the help of we can produce the new variety, we can produce the recombinant DNA, we can produce the different vaccines, we can produce different combinated varieties. Here in enzyme, there are the different types of the enzymes. It is also called the restricted enzymes because the restricted enzymes, why it is called the restricted enzymes? Because it restricts this bacteria, especially they are produced in the bacteria, and it restricts the growth of the virus. It breaks the endonuclease, especially the restricted endonuclease enzyme. It prevents or it breaks the viral DNA. And that's why it is called the restricted endonuclease enzyme, and especially it is produced by the bacteria. So there are the different types of the nuclease enzyme. Nuclease is means the enzyme nuclease which breaks the nucleic acids. That's why it is called the nuclease enzyme. The first nuclease enzyme is the endonuclease enzyme. The first nuclease enzyme is a endonuclease enzyme. This one is the exonuclease enzyme and the endonuclease enzyme. Exonuclease enzyme means suppose this is the DNA and it cut the end part of the DNA. End part. This is the end part. And here the end part is present. So it cut the end part of the DNA. Whatever may be the DNA we choose and we have to cut that DNA. The end part of the DNA, whenever it is cut by the enzyme is called the exonuclease enzyme. And the another one is the second one is the endonuclease enzyme. So the enzymes, the DNA which is selected to cut the with the help of that endonuclease enzyme or the nuclease enzyme. And when the enzyme it cut the within the part of the DNA, then it is called the endonuclease enzyme because here the middle part of the DNA it get cut. That's why it is called the endonuclease enzyme. And the exo means the end part of the DNA. And endo means within the DNA. When the part within the DNA it get cut, then it is called as the endonuclease enzyme. So do you get my point? Exonuclease and the endonuclease. Exo means the end part of the DNA and the endo means within the DNA. And how that endonuclease enzyme as well as the endonuclease enzyme it get nomenclature or the how the name is given to that enzyme. E. coli R1. There are the different types of the endonuclease enzyme. E. coli R1 is the one of that example. E stands for the Escherichia. So the first letter is chosen and it stands for that E. CO, the first two letters of the species, Escherichia coli. Coli is the species and Escherichia is the genus. So the first letter of the genus, E. E stands for the first letter of the genus and CO stands for the first two letters of the species. R stands the RY13 and first stands the type of the enzyme. There are the different types of the endonuclease enzyme. Type first, type second and the type third. So these are the different types of the endonuclease enzyme and generally type second is used in the recombinant DNA technology. The question is asked in CET also as well as in the NEET. Generally which type of the enzyme, endonuclease enzyme is used for the recombinant DNA technology. And the answer it will be you have to give the answer as like a second. type second is used. So as like this here the nomenclature is given, or the enzyme, restricted enzyme, the name is given to that restricted enzyme. There are the different types, examples of that enzymes. Alu first restriction enzyme, here the restriction enzyme alu first. Alu first, that is the arthrobacter. Arthro, A stands the genus, first letter of the genus. Lu stands the two letters of the species. And the recognition sequence is as like this here, A G C T. We have to we will learn about that, what is the recognition sequence. Then product it is produced as like this, and here the blunt ends are produced, not the sticky ends. What is the meaning of sticky ends and what is the meaning of blunt end? We will see here. The BAM H1, Ba, that is the B is the first letter of the genus, and the AM is the first two letter of the species. H stands for here the HI is the enzyme, and first is the enzyme which is to be discovered. Here the sticky end is produced. Look at here, this is the sticky, one end is here the sticky. What is the sticky end? We will see. E. coli R1, we have seen that first letter of the genus is the E. First two letters of the species CO, and here the CO, it is the second letters are the CO, that is the first two letters of the species. R is the strain, RY13 is the strain, and the first the type of enzyme which is to be discovered. Hint, Haemophilus influenza. H is the genus, first letter of the genus, Haemophilus. Influenza, the first two letter of the species IN. D stands for the strain, RD strain is chosen here, and the second means the type of enzyme which is to be discovered. And here the ends are produced, blunt ends, sticky ends, and the blunt end. What are the different blunt ends? We will see here. Now, what is the recognition sequence? Recognition sequence, we have seen that the recognition sequence, the enzyme, it cut the DNA whenever it recognizes the whatever may be the gene. Whenever you apply, or whenever that bacteria, it secretes the endonuclease enzyme, firstly, it recognizes the sequence of that viral DNA. And the recognition sequence is like a palindromes. That is the sequence of the site of the DNA, which is recognized by the enzyme. It's called as the recognition sequence. Look at here. Sequence means the sequence of DNA, which is recognized by the endonuclease enzyme. The site of viral DNA, or the site of the DNA which is recognized by the enzyme, is called as the recognition sequence or the recognition site. And it is like a palindrome. Palindrome means, look at here, this is the Malayalam. Okay, Malayalam is the here. Malayalam. So this is the Malayalam, Malayam. Malayalam is the group of letter. Malayalam is a group of letter. This is the group of letter. Malayalam, Malayam. Malayalam is the group of letter. When we read a group of letter, when we read forward as well as backward direction. So the group of letter which makes a word, which makes the same word, when we read forward and a backward direction, then it is called the palindrome. Look at here. A group of letter which makes the same word when we read forward as well as a backward direction. Malayam. Malayam. Malayalam. It is a group of letter, okay, which produce a same word. But in case of the DNA, what happens? It is the same sequence. It is the same sequence. When we read forward as well as a backward direction, this is the forward direction and the backward direction. When the orientation is kept the same, this is the orientation. 3 dash and 5 dash is the orientation. So when we keep the orientation is the same, 3 dash to 5 dash, and when we read 3 dash to 5 dash, no doubt we can read forward as well as the backward direction. But when we kept the direction is the same, 3 dash and the 5 dash is the direction, and it is when we keep it is the same, then it is called the palindrome in case of the DNA. As well as here, look at here, it is a mirror image. CTT is the mirror image of that CTT. That is the half of the first strand. This is the half of the first strand. Is the mirror image of that half of the second strand. So it is a mirror image. So there are the three different characters to recognize that palindrome or to recognize that sequence. It is as like a palindrome. How can the enzyme recognize the DNA where it has to cut? When the enzyme selected or enzyme is applied on the DNA to cut that DNA, here it has to recognize that sequence. Which are the sequence? Or the sequence is as like a palindrome. Palindrome means a group of letters which makes the same word. Then in case of the DNA, it is the same sequence. It is a same sequence when we read forward as well as the backward direction, but when the orientation is kept the same. And it should be the mirror image of first half of the first strand and the second half of the second strand is the mirror image of that DNA. So these are the different characters of the recognition sequence. So as soon as the endonuclease enzyme enter into the cell and it has cut to that DNA or it has cut to DNA, then the recognition sequence or that sequence is recognized by the enzyme. That's why it is called the recognition sequence. Recognition sequence. What is the meaning of that recognition? The sequence is recognized by the enzyme, endonuclease enzyme. Then how that endonuclease enzyme it cut the DNA? That it is called the cleavage pattern. How the DNA is cut by the endonuclease enzyme. Suppose this one is the palindrome GAATTC, and on another strand here the CTT These are the different complementary nucleotides present on the two different strands. Firstly, whenever the enzyme is applied, it recognizes the recognition sequence. So this is the recognition sequence. This is the recognition site, which is recognized by the endonuclease enzyme. Endonuclease enzyme, it recognizes here. We have to cut here. Yes, we have to cut here on that sequence, on that particular area, on that particular site. We have to cut here. In another site or in another sequence, the sequence are not allowed to cut the DNA to that enzymes. This is the site. This is the only site where the endonuclease enzyme is applied and it cut the DNA. Then what happens? Whenever it recognizes the site, recognition sequence or the recognition site, the firstly that restricted endonuclease enzyme it applied or it get cut the phosphodiester bond. It is also called the backbone of the DNA, phosphodiester bond. You know that the phosphodiester bond which is present, this is the DNA strand or the nucleotides, nitrogen bases. It is present at a first carbon atom, at a fifth carbon atom. The phosphate group is applied. Phosphate group is present at a fifth carbon atom, and here this is called the nucleotides. That is phosphate group, nitrogen bases, and whenever what we call it is a sugar, pentose sugar. This is the pentose sugar. This pentose sugar it is attached to the nitrogen base at a first carbon atom. Phosphate group is attached to the fifth carbon atom. This is the another nucleotide, and this another nucleotide, on that another nucleotide here this is the another nucleotide. This phosphate group is attached to the third carbon atom of pentose sugar, and here the phosphodiester bond is present. Here the phosphodiester bond is present. So as like this here, the phosphodiester bonds are present here. These phosphodiester bonds are present in between that nucleotides. So the firstly endonuclease enzyme, it cut it cut the phosphodiester bond, which is called as the backbone of the DNA. So firstly, it get cut the backbone of the DNA. Then it cut the weak hydrogen bonds. Here it get cut. The weak hydrogen bonds are cut with the help of that with the help of that recognition restricted enzyme, endonuclease enzyme. And by breaking that weak hydrogen bonds as well as by breaking that phosphodiester bond, here the two strands they get separate and the sticky strands are produced. These are the sticky strands. Here these are the sticky strands. Look at here. This is the sticky strands. That is, one is the short and another is here. One is here the longer one and another is here the shorter. It is called the sticky strands. Why it is called the sticky strands? Because whenever such DNA, it enters, or when we introduce into a plasmid, we have seen that during that recombinant DNA, if one arm or one the strand is a longer one and the another is the shorter. This is called the sticky strand because the whatever gene, desired gene, which is introduced into that, here it get attached with the help of that sticky strand. It gets sticks to that plasmid. That's why it is called the sticky strands. Okay. So the sticky strands are produced during the cleavage and the Mg++ ions are required for the cleavage. More Mg++ ions are required to cleavage. So these are the this is the cleavage pattern to cleave the DNA by restricted endonuclease enzyme. Firstly, it recognizes the restriction site. Then it breaks the phosphodiester bond, which one is the backbone of the DNA. Then it breaks the weak hydrogen bond and produces the sticky strands, and it requires the Mg++ ions. So for the cleavage pattern, it breaks the backbone of the DNA. Weak hydrogen bonds are also break sticky strands, sticky, and to produce the sticky ends, and then it requires the Mg++ ions. So this is the cleavage pattern. Do you get my point? Look at here again. Which are the different biological tools? The enzymes, cloning vectors, and the component host. Firstly, today we are going to see here only the enzymes. The enzymes, that is to cut the DNA. It is a biological tool. It is also called the biological scissor. It is also called the genetic scissor. It is also called the genetic scalpel because it cut the DNA. There are the different types of the enzymes. Restricted enzyme. Why it is called the restricted enzyme? Because it cut the DNA, especially the endonuclease enzyme. It is produced in the bacteria and it restricted or it restricts the growth of the virus. That's why it is called the restricted endonuclease enzyme. There are the two types of the enzymes, exonuclease enzyme and the endonuclease enzyme. Okay. So these are the two types of enzymes. Then the nomenclature as like a when the first gene or the first letter of the genus, this is E, indicates the first letter of the genus. CO, it indicates the first two letters of the species. R indicates the strain, and if first it indicates the enzyme, type of enzyme which is to be discovered. These are the different examples. Then the palindrome. Palindrome means the group of letters. The group of letters which makes the same word when we read the backward and the forward direction. Then it is called the palindrome. In case of the DNA, it is the group of sequence, same sequence when we read forward as well as the backward direction when the orientation is kept the same, as well as it is a mirror image. Then for the cleavage pattern, the restricted enzyme has to recognize the firstly the recognition sequence of the site to cleave that pattern or to cleave that DNA. Then here it cut the phosphodiester bond, then cut the weak hydrogen bond and produce the sticky ends. Then I will give a link to solve the questions. You have to solve the questions.